All right, hello everybody. Hey, I am in our laboratory today. I'm doing a little mad science, getting ready for our cell size lab. And so I thought I'd go ahead and quickly record these notes for us. If you've already seen some of the notes in class or online, feel free to fast forward ahead to the section that would be useful for you. All right, and again, as usual, thanks to David Kanufke for these notes. So let's start with some background information, a little historical information. This handsome man, Anton van Leeuwenhoek, was the first person to look at, first person to look at biological material under a high-powered microscope, which is actually a lower-powered microscope than the one that the student model that we work off. And then Robert Hooke was the first one to come up with the name cells. He looked at this sample of cork under a microscope, and the image reminded him of the small rooms that monks lived in monasteries. And so he called them cells, and that name has stuck ever since. All right, so just like molecular biology has the central dogma, fancy word for DNA's information being transferred to RNA, which then is used to make proteins. That's the central dogma of molecular biology. Cell biology, their cell theory is sort of their foundational idea. And cell theory is really obvious. All living things are made of cells. The cell is the basic unit of life, and all cells come from pre-existing cells. The problem always for students and for adults alike, we're trying to figure this one out, is where did that first cell come from? What does it look like? Where does DNA come from? All that's incredibly complicated, and science has not answered that yet. All right, we're going to compare the two major types of cells that are on this planet. They're called prokaryotic and eukaryotic. My picture was covering one of them, so I thought I'd unhide it. So prokaryotic cells, you might recognize the image. It's the one in the upper right-hand corner, and those are bacteria. They are simpler. They don't have a nucleus. They don't have a mitochondria. In fact, mitochondria are about the same size as bacteria, and that, they think, gives us a little hint about the history of mitochondria and where mitochondria may have come from. See if you can figure that out on your own. We'll learn about that later. So they're much, much smaller than eukaryotic cells, which is what's pictured below. The image is misleading, so we'll come back to that in just a second, just so you know. And then they're much more abundant. In fact, in your body, you have far more bacterial cells in your intestines than you have human cells in your whole body. That's a weird concept, right? All right, eukaryotic cells are full. They're the cells that you probably think of. They're full of organelles. They have Golgi or chloroplasts or mitochondria, for sure a nucleus with DNA in it. They're much larger, and there's two types of these cells, which we'll talk about in just a second. I'm going to move my picture for just a moment. So I added this little note that says this prokaryotic cell is actually about the size of this organelle. So that's supposed to be a mitochondria in their diagram. And in fact, even this is unrealistic. Some cells, I'll make up a number because I don't know the maximum, but I know some cells have like 900 mitochondria in them. So, lots and lots of bacteria can fit, lots of prokaryotic cells can fit inside of a eukaryotic cell. And just a real quick history of the words prokaryotic and eukaryotic. The way it was always explained to me, pro means before, kary means nucleus. So they arose before there was a nucleus. Eu means true, and kary means nucleus. So they have a true nucleus. I've never looked up to see if that's really accurate or not, but that's the way it was taught to me. All right, so eukaryotic cells, there's two types. There's the plant-like, which are called photoautotrophic. Photo refers to light, auto refers to self, and troph refers to food. So these are organisms that can make their own food through photosynthesis. They don't need to eat. That's not us. So plant cells and certain protists, these little guys off on the left, the chlamydomonas, they have chloroplasts and they're able to photosynthesize. Obviously, we don't have chloroplasts, so we are in the animal-like category. They're chemoheterotrophic. Chem refers to chemicals. Hetero means different in this case. So it just doesn't mean self. It means outside of self. And then troph again means food. So these are organisms that eat in order to obtain their nourishment. There are some organisms, Euglena, I believe, is an example, that can both eat and photosynthesize. So it's not exclusive. All right, let's just talk really quickly about the importance of membrane-bound. Oh, I didn't expect that to get bigger. Sorry, hold on. The importance of membrane-bound organelles. There we go, messing up with my name, or maybe my photo. So by enclosing parts of the cell membrane, eukaryotic cells are able to specialize. This is important. Like, I know it sounds boring, but this is a really important reason why we have eukaryotic cells. Membranes isolate areas of the cell, which allow the cell to have varied conditions in different regions. So you could have an organelle that has a different pH, and that allows certain reactions to happen in there that wouldn't be happening in the rest of the cell itself. Or we can change the concentration of nutrients in a particular part of the cell so that the digestive enzymes can work on it, whatever. And so organelles allow different reactions and different things to happen in different parts of the cell, and that increases efficiency. In addition, those inside membranes are full of proteins that do things for the cell. And so it's another surface area inside this giant cell where reactions can be occurring, and that's super important. The specialization of cellular regions is what makes eukaryotic cells much more complex than prokaryotic cells and is a prerequisite for multicellular life. Do my skin cells have the same function as my liver cells? So I need that for multicellular life. All right, moving on. All cells must do the following to survive. They must process matter. So molecules need to be acquired, and you acquire molecules through eating, through absorption, through breathing. Those are all examples of ways to acquire molecules. Those molecules need to be digested or broken down, and then we reorganize them. So if I take in carbon, hydrogen, oxygen, I can build carbohydrates and lipids. But if I don't acquire nitrogen, then I can't build nucleic acids or amino acids. And so that's a problem. So it's important that those molecules be acquired. They need to process energy. So in order to process matter, it takes energy. A lot of the energy comes from sunlight ultimately, but it can also come from food or from chemicals in the earth, especially organisms that live at the bottom of the ocean where there is no sunlight. Those ecosystems are often dependent on chemicals that are found at the bottom of the ocean that come up through our hydrothermal vents. All right, and then all cells have to process information. Most instructions for the cell come from DNA. However, signals from the environment must also be interpreted. So that's a crazy thing, that your cell can interpret what's going on in the environment and then adjust accordingly. Many cells also reproduce and communicate directly with other cells. All right, so really quickly, cell size and scale. Here's what I think is a completely unhelpful image that shows different sizes. And I don't think it's really something that any student finds useful, because it implies on the far left that an atom is this big compared to the human. It's like the size of her head. And so I get the logarithmic scale down at the bottom, but I think most students lose that perspective. So let me see if I can click on this. I think I can. Here we go. So if we go to the cell size and scale, and you can click on it independently and you can skip ahead if you don't want to see this section, but this is designed specifically to sort of show students a perspective. So if this were the size of a coffee bean or a grain of rice or a sesame seed, they're a little bit blown up right now. Let's scooch forward and see how big things are. So as we scooch in, we can see a grain of salt, and we can still compare that to the coffee bean over here. Here's an amoeba. An amoeba is about the limit of human vision, certainly beyond my human vision. I've got sucky eyes. So sometimes we can see amoeba with our eyeballs, but for the most part, that's a no. Then we get down to paramecium, which is another protist, a single-celled organism that cannot be seen with the naked eye. And then skin cells, human egg, we're getting smaller as we go. Red blood cells, notice the red blood cells, remember, are missing a nucleus because they need as much space for hemoglobin to carry oxygen. And so that nucleus gets disintegrated over time. There's the linear chromosome that we see in a eukaryote as opposed to a prokaryote. It has circular chromosomes. Moving in, notice the similarity in the size between a mitochondrion and a bacterium, and that's going to be something that we come across later. Lysosome we'll talk about in another set of notes. And now we're getting closer to the viruses. Viruses aren't considered alive at this time because they're not made of cells. So they're smaller than cells. They're just really molecules, and you can see influenza, HIV, hepatitis, rhinovirus that causes the common cold. And now we're at the level of the ribosome. The ribosome, remember, or if you don't know yet, you will, is the thing that's going to make our proteins. It's going to read the RNA, the message from the DNA, and it's going to build proteins. And then down here are some proteins that RNA, the ribosome makes. So like hemoglobin or an antibody. Here's a tRNA molecule we'll learn about a little bit later. And now we're really getting tiny. The phospholipids that the cell membrane or plasma membrane is made of. Methionine, adenine, glucose, giving you just a sense of their sizes. And then finally we get down to a water molecule and a carbon. But that just kind of gives you a sense of how much structure there is far, far, far below what a human eyeball can see. All right, let's continue on. I don't know if I can hit present on this. How do I do that? There we go. We got it. Okay, I show this video in my class. I'm not going to show this video right now, The Inner Life of a Cell. Super, super great video. I recommend coming back to it at least a couple times during the course of the unit, but we're not going to watch that right now. All right, let's start talking about organelles, or at least the structure of a cell. So cytoplasm or cytosol doesn't really count as an organelle, but it is the liquid of a cell, and it's where all of the reactions are happening. It has certain salts and proteins, but it is primarily water. So it is the medium in which all the cell's chemical reactions take place. The cell wall, we know there's cell walls in plant cells. Human cells are soft and squishy, so we don't have a cell wall. But we know that plant cell walls are made of cellulose, and we're not able to digest cellulose. If you remember, we don't have the right enzyme. So remember, cellulose is made of repeating glucose molecules, but the glucose molecules are constantly flipping. We say it's beta-linked, and our enzyme just doesn't fit that, so we can't pull those glucose molecules off of cellulose. Bacteria, their cell wall is made of peptidoglycan, which is another carbohydrate. But what's interesting is because bacteria have that in their cell walls and humans don't, we can take medicines, antibiotics, that mess with peptidoglycan, and it won't hurt us at all because we don't have peptidoglycan. So it damages the plants or damages the bacterial cells. And then in addition, fungi, their cell walls are made of a molecule that's called chitin, which is the same molecule that insects use for their exoskeleton. And again, for drugs like antifungals, if we have a fungal infection, we can take drugs that mess with chitin. And since we don't have chitin in our bodies, it just messes with the fungus and not with us. Pretty smart. All right, cell membrane, we're going to learn lots more about cell membranes as we go. So it's a phospholipid bilayer with embedded proteins, and you really are going to get to know this entire structure. I will tell you that there are scientists who've spent their entire lifetime studying cell membranes, which to most students is like, you can't even fathom that. The reason that would happen, I'll just give you a couple examples. Cancer cure is so scientists who are scientists who are studying cancer, who are trying to get rid of or destroy cancer, or stop cancer, will look at cell membranes and see if there's a way, this is just one example, to tag a cancer cell so that the immune system wakes up and identifies it as a cancer cell and eats it. There's other scientists who are studying the cell membrane to understand HIV. HIV enters through one of the embedded proteins, and is there a way to block that protein so that HIV can't come in and is never disruptive? Those are just a couple of examples, and those couple of examples have the potential to change the world. So the function of the cell membrane, if we're being very general, it controls the transport of matter into and out of the cell, and it receives and sends messages within the environment. This is a transmission electron microscope photo of the cell membrane. It's really hard to tell, but this line up here, those are the hydrophilic heads. And then in between are the tails, the hydrophobic tails. And then on the heads are down at the bottom again. So we call over here, above the cell membrane in this diagram, we call that the extracellular fluid. That's whatever the cell is floating in. And then on the inside, notice cytoplasm is marked. All right, I'm going to pause my video for just a second because I need to do something with the lab that I was doing. So give me just a moment. I'll be right back. Okay, I'm back. Unfortunately for me, they weren't ready, so I still have to, I'll do a little more of this presentation and then we'll be able to come back to it. All right, next organelle that we're going to talk about, this is really an organelle now, is the nucleus. Of course, everybody knows the nucleus is the place where the DNA is stored. It's a double membrane with protein pore channels. I got to set my keys down here. And so I really want you to notice the nuclear surface has these pores on it. There's a picture of it over, point that way, there we go. And those are the pores. If you watch the inner life of a cell, you saw the nucleus. It looked sort of like the surface of the moon, and then things were shooting out of it. Those things that are shooting out of those pores, that's the mRNA. And then the ribosomes are grabbing onto it and reading the mRNA and then building a protein. So it's the site of DNA storage and replication, which means copying the DNA so that the cell can divide. And it's an information relay network to the ribosomes. And that's what we were just talking about. Okay, the nucleolus is that dark spot that's in the nucleolus, and it's where our ribosomal RNA genes are concentrated. So basically, it's the mother area of our ribosomes. So it's going to tell the body how to build ribosomes. The thing that's tricky about ribosomes, I think they're next here, ribosomes are actually made of RNA that's kind of twisted on top of itself. So it's coming from the nucleus. The instructions for building those ribosomes is coming from the nucleus. So it's a complex of RNA, which we're going to learn is called rRNA. The little r stands for ribosomal RNA, and protein. It has two subunits, the large subunit and the small subunit. And then what's running through it is the mRNA. And the ribosome is reading the mRNA, the letters, remember A, U, C, and G, that's on the mRNA. It's reading those letters and it's saying, okay, this is the protein that the DNA wants to build. We'll learn how that happens in detail very soon. Eukaryotic ribosomes are larger than prokaryotic ribosomes. That's just something that's good to know. And get my image out of the way here. And it says the site of protein synthesis. So here's one thing that I know was confusing. One of my students mentioned that in the Biozone ebook that it said that rough endoplasmic reticulum was the site of protein synthesis. Technically, both are correct. The ribosome is actually synthesizing the proteins. That's what it's doing. The rough endoplasmic reticulum has some ribosomes embedded in it, and they're doing protein synthesis. So the rough ER is a place where protein synthesis is occurring, but the rough ER itself is not doing the protein synthesis. It is the ribosomes that are doing it. Okay, so now we're going to talk about the endomembrane system. That'll get us through a whole bunch of the organelles. It starts in the nucleus and it heads all the way out to the plasma membrane, and we're going to talk about that. All right, so there's two kinds of ribosomes. The first kind of ribosome is called a free ribosome. This photo, again, is a TEM, so transmission electron microscope, which allows us to see much, much, much smaller things than a light microscope does. So the polka dots that are floating not attached to the lines, those are called free ribosomes, and they tend to make the proteins that stay in the cytoplasm. However, the bound ribosomes, those are the ones that make rough endoplasmic reticulum look rough. They're attached to the endoplasmic reticulum. They are making, and I think that they can switch. I think sometimes they're on the ER and sometimes they're not. But they are the ones that are making proteins that go into membranes or are exported from the cell itself. Ribosomes can become free or bound. Look, I have it right there, based on the protein that they are making. All right, and then endoplasmic reticulum. There are two types of endoplasmic reticulum. What is it exactly? My picture's in the way. So it says a network of membrane channels attached to the nuclear membrane. Rough is closest to the nucleus. So take a look at the purple nucleus in the image. It's covered in ribosomes. Smooth is farther, and it doesn't have any bound ribosomes. In this, it looks like a series of tubes or something. Hard to describe that. Okay, so what's their function? The function of the rough endoplasmic reticulum. Again, this sounds boring, but the compartmentalization is super important. Having an area where certain reactions can occur separated from the rest of the cell is really important in a eukaryotic cell. So it compartmentalizes the cell, it provides structural support and targeted protein synthesis. So those bound ribosomes are doing very specific protein, making very specific proteins. And then the smooth endoplasmic reticulum has a whole series of different functions. It synthesizes lipids, so that's where our our fats are being made for things like membranes. So like the phospholipid bilayer, I should say. It detoxifies poisons or things that are bad for the cell, and it breaks down glycogen. If you remember, glycogen is also a carbohydrate that's made of a whole bunch of glucose molecules. It branches. It's got a different structure than cellulose or starch, but the cell is able to pluck those glucose molecules off for then use so that we could use those in our mitochondria. Okay, vesicles. Vesicles, there are three main types of vesicles that we're going to talk about. They are a small compartment surrounded by a phospholipid bilayer. Really, all these things are surrounded by phospholipids. So they have various functions depending on what's inside of them. They're like little storage molecules. So they're transport molecules or transport vesicles. So let me show you down here. So the ribosomes made proteins, and then they packaged them, the smooth endoplasmic reticulum packaged them into small vesicles that then meld or merge into the Golgi apparatus, which is that series of sort of green pancakes there. So that's a transport vesicle. The secretory vesicles, they allow the cell to secrete. So I'll get my image out of the way here. Trying to get my image out of the way there. There we go. Secretion is happening up at the top of this image, and you can see molecules coming out of the cell. Those are proteins that the cell made that it's releasing out of the cell, or other molecules. And then lysosomes, you can see a lysosome right here. Lysosomes we'll learn more about in the next presentation, but lysosomes have digestive enzymes in them, and they break down molecules or old cell parts. They even help a cell to suicide if the cell is malfunctioning, if it's not working properly anymore, or if it's gotten old. All right, the Golgi apparatus, oftentimes called the Golgi, gosh, I got to think of all the words, just the Golgi, Golgi body, Golgi structure. There's a bunch of names for Golgi. So if it's Golgi, it's Golgi. So it's a series of flattened membrane-bound sacs. And just to remind you, it was right here in this previous image, right in the middle of the previous image. Its function is to take things that come into it and process them. So let's say a protein comes in. It's going to maybe add some different tags, some different molecules to the protein to make that protein just right. We'll learn more about that coming up. It then packages them in vesicles that then it can secrete out of the cell. And so this image, the cartoon image, is sort of upside down. They're coming in from this little vesicle here. They're melding with the Golgi body. They're being in this cisternae, the middle part of the Golgi is where they're being modified and molecules are being added to the protein or whatever molecule it's working on. And then once it's ready, then it gets placed in another vesicle that then melds with the cell membrane and is released out of the cell. So this is actually our last slide. I've included a couple of videos that I think are really good. The first one, oh my gosh, it's 1 minute long, totally worth your time. It just shows you from beginning of endomembranes, from the endomembrane system all the way through to the end of the endomembrane system. And it really pulls into perspective all these things that you're like, oh my gosh, what is she talking about? It simplifies it so much, and it's a good use of your 1 minute. So it talks about exocytosis and endocytosis. And I was telling you about how the cell can secrete things from the cell. So exocytosis is secretion. It's the discharge of a vesicle, whatever was inside that vesicle, into the extracellular space, into the space on the outside. Endocytosis is the exact reverse process. Endocytosis is when the cell membrane, I can't do it with my fingers, but it pinches in and then makes a vesicle. And so it's bringing something from the outside in. And so it's like it could eat, for example. It's not really eating, but it gives you an idea of that. And then there's one other video here that you could watch. This one's about 3 minutes long, and I recommend it. But if you're going to watch a video, for sure watch the first one. All right, ladies and gentlemen, that is it. Let me know if you guys have any questions. Have a great day.